Today I'll be presenting my research that I conducted with Dr. Wallace on the role of PMK-1 gene in cell migration. And our lab is very interested in this process as it is important in many biological processes. For example, if cell migration does not occur properly, it can result in diseases such as cancer, as shown in the figure. So here we have a representation of skin cells that fail to migrate properly, and they eventually result in tumor growth. Furthermore, cell migration plays key roles in the morphological process of many single and multicellular organisms with the aid of encoded genes and proteins. And what we wanted to do was we wanted to find cells, genes that potentially play a role in cell migration. So we know that the wave-scar pathway controls cell migration in C. elegans as illustrated here. And what we have here is 3 axonal guidance receptors on 40, SAX3, and VAB1 that control cell migration in C. elegans. So these receptors, they send a message to the GTPase SED10 RAC1, and this GTPase acts as a switch to turn on the wave complex, illustrated here, and it activates the wave complex. These genes activate the wave complex. In particular, what I would like to point out is the GEX genes. Now, why is it called GEX? It's called GEX. It stands for gut on exterior. And this is due to the mutations that occur in the genes, and it prevents migration from occurring properly. So the skin of the embryos is not able to seal, and therefore the gut of the embryos, it comes through the skin, and we eventually end up with a dead embryo. So, however, if the activation of the wave activation of the wave complex does occur normally, it makes branch actin, which drives cell migration. So we use C. elegans as a model organism to study cell migration, as its genome has been sequenced in its entirety, allowing for easier manipulation of its genes, and they mirror the mammalian system, among other components. So we use the process known as RNA interference to screen for genes that may potentially play a role in cell migration in C. elegans. And RNAi is basically a mechanism for silencing genes or silencing gene expression using double-stranded RNA. So what we have here is a general process of an RNA screen. We used a similar protocol in our lab. The only difference was that we used the feeding RNAi protocol. So instead of injecting and incubating the worms in the RNAi, we fed the RNAi to the worms. And from this screen, we found our gene of interest was to be PMK1. So I constructed a domain structure to find out what functional regions of the protein, And I found protein kinase ATP, MAPK, and STKC P38. And the processes shown here the processes shown here, they're known to be involved in the mammalian system. So we have functions such as cell proliferation, regulation of stress and heat responses. But in our study, we wanted to show that it functions in cell migration and how that occurs. So one of the domains we found was the mitogen activated protein kinase, or MAPK, and this domain is responsible for cell growth and cell differentiation. Another domain was the protein kinase ATP, and that is responsible for mainly protein phosphorylation, and it aids in cell proliferation as well. And finally, the other domain that we found was the serine threonine kinase, P38 mitogen activated protein kinase. And this also plays regulatory roles in cell proliferation, and it also mediates immune response. So in humans, PMK1 is homologous to the gene MAPK14, and we found similar functions as the PMK1 domain. The only difference was in the STK, well, slight differences in the STK CP38, and that was the fact that they respond to external signaling and dopaminergic signaling. So we know that mutating PMK1 results in dead embryos. So in the left panel, we have a representation of a wild type, or what we deem as a normal embryo. And here the embryo is in the twofold stage of development. So it's beginning to form into a worm. On the right, in the right panel, this is at the same stage, but here you see that the embryo is flattened and it fails to develop properly, which eventually results in a dead embryo. So to find out where exactly PMK1 fits into the wave-scar pathway, we first checked it on the axonal guidance receptors I mentioned earlier, SAX3, VAB1, and 40. So what we found was that, so on the y-axis, we have the percent abnormal embryos or dead cells. And possibly this is due to the fact that there's lack of proper cell migration. So in T. elegans, as I said, N2 represents the wild type, and here we have 0% death. So when we introduced the PMK1 RNI on the N2, we found that there was over 20% death, a death toll of the embryos. So also again, recall that we tested on the axonal guidance receptors VAB1, SAX3, and 40. For VAB1, we saw about a 40% death, but when we introduced the PMK RNI on VAB1, it went further than the 20% death toll that we saw in N2. So we saw about an 80% increase in the percent abnormal embryos or dead embryos. SAX3, we didn't see as much as for VAB1, but there was still a higher percent of abnormal embryos presented. The one we saw little to no change was the 40 mutants, and the one we saw less change was the 40 mutants. So what does these results mean? So basically, when two mutants enhance each other, it means that they're functioning together in parallel pathways. As in the instance of 40, where we didn't see much of a change, we're suggesting that they potentially function in the same pathway. So to just reiterate what the results I found from my research. So we first used an RNA screen to find the gene PMK1, and it potentially plays a role in cell migration. The second thing was that we figured out that it may be functioning in a similar pathway as one of the axonal guidance receptors on 40. We also realized that the embryos that lose PMK1, they do not develop properly, and they're instead flattened as shown in the figure, and they eventually die. So we got numbers such as I said, 20% of the time the embryos died. And finally, we looked at functional domains of PMK1 gene, and we compared it to the human MAPK14 gene. So we found similar functions between those genes, and we hope to further investigate these similarities. So essentially, what we want to do, we want to determine how PMK1 affects other components of the wave pathway, and we want to measure actin levels in PMK1 mutants to further understand how PMK1 affects the process of cell migration. As I previously stated, actin recruitment is required for cell migration. And I would like to thank Dr. Wallace for helping me with my research, the FDU Bio Department for giving me the opportunity to conduct research, and I would like to thank GSL SAMP for funding my research. Thank you.